Hey, good evening, everyone. This is Kanika Gupta, TA for the session, and we'll be taking classes every Tuesday from 6 p.m. to 8 p.m. And we'll be discussing the previous week's lectures, and then we'll discuss the assignment questions. And I'll bring some questions, and we'll discuss that. Meanwhile, if anyone has any doubts, if anyone wants to ask anything, they can ask that in between the lecture, after getting the lecture completed, whenever you want. There's no issue on that. You can ask your questions in between also. So every time our lecture will proceed that way only. And if you guys have any issues in the lecture, you can tell me and we can improve on that. Okay. So we'll first go through once what you guys must have studied. I'm just hoping that you guys have listened all the videos of previous lecture of week 1, and we'll be discussing just a quick recap of what was there in week 1's lecture. Okay. I hope my slides are visible to you all. If any one of you can give me a thumbs up. Perfect. Thank you. Uh, Before starting, I just want to know if you guys are comfortable in telling me how many of you are from biology background, who know the biological terms. Shalini is from biology background, other people. So most of you are from biology background. Perfect. Great. So let's start then. So yes, what is developmental biology? This course is basically will be telling you about what is developmental biology, how things happen during the development, what are the different kinds of cells, what are the different kind of changes that are involved in forming a particular organ or the whole organism. So there are many questions that will be answered during the course. So and I would request you, being a biology student personally, that please do not miss any of your lectures, so that you won't miss the link, because biology is, according to me, according to my personal experience, is a subject in which if you forget the connection, then you won't be able to pursue further. You won't be able to understand कि जो अब next हम बता रहे हैं, what does that mean, how does that links with the previous one. So I would request all of you, do not miss your lectures at all, because that will help you in linking your previous knowledge to the one that you are studying now. So it will help you in that ways. Otherwise the subject is very interesting, and I hope you guys also enjoy taking the lectures and attending the doubt sessions. So yes, uh, according to molecular biology, we had the answer for genetic material, we had the answer for central dogma, basically how the things, how the proteins are formed, from the information came, what is the genetic material, how the information is transferred from genetic material in the live molecule in the form of a protein, in the form of different macromolecules, who is giving the instruction that yes, this process has to occur, and these are the molecules that will be involved in the process and everything. So we've got that much of idea from molecular biology, which says that the DNA is the blueprint of an organism, basically the genetic material, which has all the information. All means all the information. Okay, then the mRNA, the messenger RNA, is formed from DNA, which is a transcript from DNA, which takes the information from DNA to convert it into the form of the protein. For that, it will require ribosomes and tRNA, the transfer RNA, and then that will be converted. The information on mRNA will be converted into the form of a protein, and that protein will further lead to many biological functions that it is supposed to perform. Okay, so this much we have understood from the molecular biology already. So, before going into the detail of developmental biology, there are some famous sayings for the developmental biology. Ki what is the basically the question of developmental biology come from ki what is life and how it is developed. This is what we are basically trying to answer in case of the developmental biology. How a single cell is able to form a complex organism. That is what the process through which a single cell can form a complex organism is what we will study in developmental biology. So, what Brenner said, all the classical problems in molecular biology have either been solved or will be solved in the next decade. The army of large number of American and other chemists into the field will ensure that all the chemical details of replication and transcription will be elucidated. And I have long felt that the future of molecular biology lies in the extension of research to other fields of biology, notably development and the nervous system. Why it is said so? Because you get the information that DNA is the one which is having all the information. But where this information is going and which cell is forming what, which cell will have a fate to form the neural cell, which cell will have a fate to form the skin cell, which cell will help in the formation of heart, which cell will help in the formation of muscles. So all of this information will come from what? Developmental biology. DNA will tell you that this is the protein, this is the molecule that will be involved, but on which cell or where it will be involved, what part of that single cell will form this complex part of the organism, that all questions will be answered from developmental biology. Okay, so what are the basic questions of developmental biology? How does fertilized egg become adult body? As I told you, how does the adult body produce another one? Okay, so these all are the different questions that will be answered in developmental biology. For example, we say ki how adult body produces another one, that comes from cellular differentiation. And obviously the answer for how does a fertilized egg becomes adult body also comes from this answer only, that the cellular differentiation, a single cell divides and give rise to different kinds of the cell, that those different kinds of the cell will again be differentiated on the basis of the different signals that it will get, that will you guys will study further in the course. And then when these different kind of a cells are formed, then these different cells will give rise to further differentiated cells, and those differentiated cells will help in the formation of different things, that different organs, different parts of the body, different kind of veins, arteries, blood vessels, everything that is involved inside a complex organism, those different cells will help in the formation of those different parts, which will further lead to the formation of a whole complex organism, or we can say an adult body. Okay, so all of this will be done under the influence of under the influence of different kind of a signals that a cell gets, and further the cells differentiate, which leads to the cellular differentiation and formation of complex organism. Next answer is the morphogenesis. Stem cell is basically a cell which is not a differentiated cell. It is a basic cell which can form any kind of a cell from itself, depending upon the what kind of a signal it is getting. Okay, so a single cell will divide, as I told you earlier. It will divide. It will be differentiated on the basis of the chemical use, environmental use, and many kind of signals involved. And then it will form a differentiated organ or a part of organ. Next answer is the sorry. Yes, next answer is the growth. Again, the developmental biology how a single cell forms the complex organism, the question still lies there. So growth is again an answer for that. The organism will grow, the cells will grow, the overall organism will also grow, reproduction will happen. That is how an adult organism is able to produce a similar one, a similar organism just like itself. So again, how is cell division tightly regulated in the growth? Obviously it is not like ki every cell will grow in any way it wants. No, it can't happen. If it will happen, then it can lead to many kind of problems inside the organism. So it must be regulated in a particular way. And reproduction also, it how the germ cells are specified, what instruction in the nucleus and cytoplasm that allows them to form the next generation. So obviously, all of these questions of developmental biology will be answered throughout the course. Evolution, how evolution is involved in formation of an initial organism and changes happening inside that organism according to the environment, according to the different kind of use it gets. So according to that, how evolution helps in shaping an organism that it is now. So again, and then environmental integration is also involved in making the organism. As I told you, environmental factors are very much involved in making an environment how it is. For an example, if you will go with a polar bear, so it has very thick fur on its skin. That is because it lives in that kind of an environment where it requires that. So that is, we can say, is a kind of an environmental hue. Depending upon that, it has evolved and has developed that thick fur around its body so that it can survive in that scorching cold. Okay, so approaches to study developmental biology, it can be anatomical, it can be experimental, it can be genetic. That we will see again further in the course. Now, the one thing that comes that is very important for you guys to understand while studying developmental biology is what is life cycle. So, life cycle is a central unit in biology. Then evolution becomes the alteration of life cycles through time. Genetics, the inheritance mechanism. Obviously, genes are the one which is transferred from one organism to another, which takes the information. And development, all the changes in structure that takes place during one life cycle. So basically, life cycle involves all of these aspects. Okay, so life cycle is basically, if we go by the simpler terms, it is that you can say the period of a person's life from the day it has taken birth till it becomes mature enough, or we can say adult enough, to give rise to a similar kind of, we can say, to give rise to a progeny. So that is what a life cycle is. So, if we'll go by the developmental history of frog, the Rana pipens, basically the leopard frog. So, obviously, the sperm and the oocyte first they fused. They came from an adult, a sexually mature adult, and when they fused, they gave rise to a zygote. And that zygote then underwent many different kind of stages of development: the morula, the blastula, gastrula. And then after these different kind of developments, reaching a specific kind of stage where it can now produce different kind of organs required for that organism, then it underwent organogenesis, and then again underwent the different larval stages to attain a particular growth, to give itself that growth period so that it can again become a sexually mature adult, and then that sexually mature adult can give rise to further of its kind. So this is the whole life cycle of what, of this frog. This happens in all the organism. It is again that period of your life in which that from the day the uh, gametes are released till the day you become sexually mature adult, is the whole period is called as what, the life cycle of that organism. So obviously, the early development of frog, they have shown these things. कि कैसे अर्ली डेवलपमेंट जो है बेसिकली व्हाई दे हैव चोजन frogs for the study, because if you guys listen the lecture carefully you must have known that this brownish part in the egg shows the animal pole, and the whitish part shows the vegetal pole. So what is animal pole and what is vegetal pole in an egg is, vegetal pole is basically the yolk part of the egg, and animal pole is basically that part of the egg which makes the actual organism. Okay, so it you can understand that for the basic knowledge. Obviously when you'll study further in the course you'll get to know more about animal pole and vegetal pole, but for now you can understand it that way, that animal pole is that part where the actual organism is forming, and vegetal pole is that part where the yolk part of the egg is laying. So it just shows ki how the simple eggs undergo different kind of stages. It undergoes cleavage, and then after cleavage at the end it forms the blastula, and then that blastula again forms the gastrula, then organogenesis, and then again a mature organism. As you can see it in the bee structure, basically in this it is showing the mating part, but again these are the sexually mature adults which will be formed after the different kind of stages of development. Okay, so what fertilization accomplishes, it accomplishes first the pronuclear fusion, basically the fusion of the gametes and then the nucleus of the gametes, then the cytoplasmic migration, then the activation of molecules necessary for cleavage and gastrulation. As I told you, there are many molecules involved, many kind of hues involved, and they should be released timely. It's not like ki once it is released it is released. No, it must be released on its time only, otherwise the total development will be hampered, and the deformed organism will be formed that may not be able to survive even. So it must be done at its particular time only. Now what happens in some organisms, the environment decides that what kind of either the male or female, what kind of an organism, what sex of that particular organism that egg will form. So that will be dependent upon the environmental use. So it happens in case of some organisms. So again the environment will the environment will affect that, and then those kind of hormones will be released in that particular zygote, or we can say that particular organism, and then it will lead in in that direction, in that particular direction, either male or female. It will lead into that direction of development, and then it will release either the egg if it is female, and sperms if it is male. So that will be depending upon the environmental use it got while it was developing. Okay, so again the as I told you, these are the different stages it goes through. The dorsal blastopore lip, you will come to know these things, all of these details when you will study further developmental biology. I won't go into the details, otherwise you'll be confused. So we'll study them once we'll go through them in the lecture series. Okay, one thing more here is, now when this structure is formed, when we get this structure, it has, if you can see, three different layers, three different colors of layers. So basically blue one is your ectoderm, pink one is your mesoderm, and yellow one is your endoderm. So basically all three of these layers are important in the formation of different kind of structures that are important for an organism, that will make the different organs in an organism. Ectoderm will be involved in the in the formation of the outer epidermis of the skin. The inner dermis will be formed by the mesoderm. I'm not sure, I have to check that, but I think it will be by mesoderm only. And then the neural part also will be mostly formed by the ectoderm part. So again obviously these different kind of structures will be formed by these three germ, इनको हम कहते हैं germ layers. ठीक है, these three are called as the germ layers, because these are the germinal layers which will help in the formation of all of the organisms inside the body of that particular Just a moment, guys. I have to take this call.
Hello.
Yes, sir.
Yeah.
Yes, sir. No, sir, I am in the session. Actually, I have put it in the presentation form, so I was not getting the. That's why I actually didn't. So I'll just. I have admitted you. Yeah, I have admitted you, sir. Yeah, yeah, I am in the session. Yeah. Okay, sir. Sorry for the interruption, guys. So let's again move ahead. So as I was telling you guys, these are the three germ layers which will form all of the organs of an organism, which will help in the formation of the complete organism. So again, the ectoderm, the endoderm, and the mesoderm. So you guys will uh, keep this thing in mind. We'll further discuss in the lecture series ki which layer forms what part of the organism. But again, these are the most important layers for any kind of zygote, any kind of a developing organism or zygote, we can say. Next is the again these are the things just to show you that how are the different developmental stages are there. And if you can see it here, this is a larval stage of an organism, and mostly it is a tadpole of a frog. Yeah, it is a xenopus leavis. So basically it is a tadpole of a frog. If you can see it, there is a tail part inside in the tadpole, which will be further lost when it will develop into a complete organism. So again, these are all metamorphosis thing that you will also study in the lecture series further. So metamorphosis is also a part of the developmental biology that we will further see in the lecture series. Just a moment. Yes, as you can see it here, this is the notochord of the developing larva, and this is the developmental stages of the frog. If you can see, a vestigial tail is still there, which will be further regressed when it will become a sexually mature adult. But these are the different developmental stages of a frog, that how it from a tadpole it becomes a complete adult frog. Next, the anatomical approaches to study the developmental biology. So it has again the four things: the comparative embryology, the evolutionary embryology, teratology, or mathematical modeling. So what happens is in comparative embryology, you will compare the similar organisms so that you can understand how the development is happening in these different organisms, and till which part they were similar and at what point they diverged from each other, either, or at which part they converge together, so that you can understand where from the ancestors are coming. That you will understand in the evolutionary embryology, when you are saying about where from the ancestors are coming and from which branch that organism has developed, and what kind of a changes the organism has faced from the ancestors till the to the organism that is it is now at present. In teratology, basically there are some chemicals which hampers the growth of the embryo and which can lead to some kind of the abnormalities in the embryo. So for that, the teratological studies help to understand which part is getting affected by the by which chemical, and which chemical is affecting which particular cell, which can lead to that kind of abnormalities in the embryo. And mathematical modeling also helps in understanding the fate map, that which cell is forming what kind of organ. So obviously we'll go into details of all of these things as we will proceed further in the lecture. In comparative biology, the term that you will listen more often is the oviparity, viviparity, ovoviviparity. So these three branches are totally depending upon that either organism is laying eggs, either they are giving birth to and whole organism, or the organism is laying birth is laying egg also, but Also, along with the laying eggs, it is taking care of its younger ones also. So that comes under ovoviviparity. So in oviparity, basically, this category involves those organisms which lay their eggs and then move ahead. Then in viviparity, this includes those organisms which give rise to which give birth to a younger one. They do not lay eggs, they give birth to the younger one. So basically humans comes under viviparity. And in ovoviviparity, most of the organism, it includes those organisms which lay their egg but also take care of their eggs till the eggs hatches and give rise to the younger ones. Okay, then again, two further categories are also under which the comparative embryology studies. It is the holoblastic cleavage or the meroblastic cleavage. So this is basically, as if you guys remember, we were discussing about the different kind of cleavages that zygote undergoes to form the blastula, the gastrula, and then the different germ layers which will further help in the development of the whole organism. So this is those kind of cleavages only, that whether it is a holoblastic cleavage or the meroblastic cleavage. If a holoblastic cleavage, a zygote from an organism is undergoing a holoblastic cleavage, that means whole of the zygote is undergoing the cleavage, not some part of the zygote. But if it is a meroblastic cleavage, then in that case it the not the whole zygote but only some part of the zygote will undergo the cleavage. So again, these terms will be cleared as we'll move further in the course, along with the examples, so that you can understand it better. Then again, the microscopic studies further helped in understanding these kind of developments. I don't know how many you have studied zoology, but if you guys have studied zoology and if you guys have attended the some of the zoology sessions, the practical sessions basically, so in that we used to do that. We used to take a chick egg and then we used to perform the experiments on chick egg just to see the development of the chick egg. So again, you can see it clearly in the chick egg, how these kind of you know from a egg, how the complete organism is getting formed, how different vessels are formed, and every part of the development you can see, because again it is totally dependent upon ki in how much time you will see the egg. This is a continuous process, this is going on and on. So again, you can see it through microscopes also, and from the naked eye if the embryo is large enough. Then initially, when developmental biology was very at its initial stage, it was thought that all the structures are prefigured in the germ cells. That means they are already there, formed in the germ cells, and when these germ cells will fuse, the structures will come along and it will form the whole organism. It was a very very early thought when developmental biology was being studied, when it was considered to be being studied. Just a minute, guys. I think no, I didn't got any notification. Yeah. So yes, this was that thought, but no, obviously it is not the case. It is the genetic material which take contains the information, which further helps in passing on the information. Um, for an example, if we'll go by the regeneration in hydra, so it has the information but it does not have a preformed structure. In hydra it has the information, and hydra will form whenever, if you guys have done the experiment, if you cut any part of the hydra it will regenerate that part. So it has the information, but it is not like it has the structure preformed inside it that it will push out whenever we'll cut the hydra. So there is a force, that developmental force, epigenetic development from preformed instructions, that Blumenbach has said that it is not the structures are formed already inside the germ cells, but it has an information, a preformed instructions which help in the generation of the complete organism. Again, the all the information that is already stored inside the organism. Again, as I told you, the germ layers, the ectoderm, mesoderm, and endoderm. Ectoderm, outermost layer of the embryo. It helps in the development of brain, nerve cells, surface layer of the skin, as I told you already. It helps in the formation of the epidermis, the nerve cells, and the brain, most of the neural part. The mesoderm, the middle layer, the pink one that I showed you, it helps in the formation of a heart, blood, kidney, gonads, and bones. Endoderm is the inner lining and it helps in the formation of the intestine, lung. And you will study all of these developments and all in the when we'll move ahead in the course. The induction for these things, that which layer will form what and which part of which layer will form what, is again comes from induction. These are all of the internal hues, internal signals that tells that you have to form this, you have to form this, and you have to form this. So it is all stored inside that single cell, which further divides to form a multicellular organism. Okay. So induction is basically interaction among these layers produce many organs that leads to the uh, basically the formation of different parts. Again, it proves that part that it is not pre-formed organism that is present inside the germlet. It is the instructions that are present there already, which will help in the development of the whole organism. As we have discussed earlier, that first zygote will form. This zygote will structure is a bit wrong. The sperm should not be here, because now the fusion has happened and zygote has formed. Then it will lead to the formation of the blastula. Then that blastula will form the gastrula, and then after that gastrula, because in gastrula we finally get the ectoderm, the mesoderm, and the endoderm. Now the function of the germ layers has started, and now these germ layers will start forming the different parts, or we can say the different organs of the organism. The outer surface, the epidermal cells of the skin, the central nervous system, the brains and the neurons, and the neural crest, basically the melanocytes and all the cells. So these structures will be formed from the ectoderm, the outer layer, the outer germinal layer, the germ layer. Then again, the mesoderm or middle layer will lead to the formation of the notochord, the bone tissue, the tubule cells of the kidney, and the red blood cells and the facial muscles. So these structures will be formed by the mesoderm. We'll go into the details again, as I'm telling you again and again, when we'll move ahead in the course. Then the endoderm, the internal layer, that will form the germ cells, the gonads part, and also it will lead to the formation of the digestive tube, the thyroid cell, the pharynx, and the respiratory tube, basically the lung cells and other alveolars. And the alveolar, basically the alveolar cells. The lung cells are the alveolar cells. Now there are some principles, basically the four important principles of developmental biology proposed by von Beers. And what did he said? The general features of a large group appear earlier than a specialized feature of a smaller group. So all vertebrate embryos have gill arches, notochord, spinal cords, and primitive kidneys. ये सबके पास होते हैं. But after a particular stage of development, the specialized feature of a particular smaller group will start appearing. But the generalized features of that a large group of organism in which the initial developmental stages are same will appear earlier. Okay, the specialized structures of a particular organism from that large group will start appearing later. Less general characters develop from more general characters. Skin development in vertebrates. Okay. Next is the embryo of a given species, instead of passing through the adult stage of lower animal, departs more and more from them. So, what does it says is the embryo of a given species, instead of passing, basically what happens is initially when the development started in a large group of organism, it was similar. It was similar till a point. Now when the different organism will develop into the different kind of organism from that particular stage onwards. It is not like that that organism will first pass through the adult stages of another organism, another similar organism but not same, and then it will develop inside into its own kind. No, it is not like that. Till a particular stage they will develop all same. After that they will depart along their particular line, particular type of development that that organism actually carries. Okay, so early embryo of a higher animal is not like a lower animal, but like its early embryo. So these principles were explained very good by sir in the lecture series. I hope you guys understood. Still, if you guys have any confusion, we can discuss that later. Next is the evolutionary embryology. This was all comparative embryology we were studying. Then the evolutionary embryology, in which we'll see that if the function of a organism of a particular organ of an organism is similar or not, or the ancestry of the development of that organism was similar or not. So in evolutionary embryology, uh, evolutionary relationship is more obvious at embryonic or larval stage than the adult stage, because according to the life of that organism, it may develop some differences in its organs according to again the lifestyle. But initially when the development started they were all similar, because again they were all forming a hand. But that hand can be a wing for a bat, but it can be a hand for a human. So again the functions are different. That is why later in the development they became different. Initially when they started developing it was all same. So that is what they want to say. So embryonic similarities means common origin, adult variations that can happen depending upon the life cycle of the organism, and developmental changes enabling adaptation. Because, as I told you, because of their different lifestyles. If you see in barnacle and shrimp, initially developmental part was similar, but when they became adults, so obviously the structures changed according to their particular lifestyle or according to that particular organism. Developmental changes is the what which enables the evolution. If you can see that the lifestyle, again the niche in which the organism is living, either whether it is terrestrial or whether it is aquatic, whether they need to eat whether they are herbivore, whether they are carnivore, what kind of food they are having, what kind of a niche they are living in. Depending upon that the changes happen, and that leads to the evolution of an organism. If you can see here, basically this colors here actually show you that the bones are similar, but depending upon the organism, depending upon its usage in the organism, the structure has become different. If you can see here in the B part, in the vertebrates basically they are showing that this upper jaw, or we can say the maxilla, how much it is different here, how it is different in human, and how it is different in an crocodile. So again the usage of the organism, these things became different. And again, as I was telling you, the human arm and bat wing, or we can say a bird wing, or we can say a sea limb. So the usage of hand in human and the usage of a wing in bat, both of these structures are hands only, but depending upon the usage of hand in human and wings in bat, they have developed differently in later stages of the development, which led to the evolution in that particular lineage, we can say, or we can say in that large group of organisms. Next is teratology. So teratology, as I told you, these are some of the chemicals which help us understand better that which affect to which part of the developmental of the developing embryo can lead to what kind of a defect in the organisms. So if you can see, this is piebaldism. This syndrome is due to mutation in the gene KIT. So animal models helps us to investigate underlying molecular mechanisms in this. So KIT is expressed in the neural crest cells, which give rise to the pigment forming cells, the basically the melatonin cells, and ear cells and the gut neurons. Also, it is precursors of the blood cells, and it is a precursor of it is involved in the primordial germ cells also. So if you can see that in the human and in the rat, both in both the cases the depigmentation has happened, because again due to due to this KIT gene was mutated, or KIT gene was affected, which was responsible for the melatonin forming cells. Then ears also got affected in these organisms, because again the kit was affected. And the gut neurons, the neurons present in the gut, or we can say the intestine part of the digestive system, also got affected. So obviously told us what is the role of that particular cell lineage. Then this person, again, the thalidomide was a very famous drug consumed by the pregnant ladies. Although doctors prescribed it for, I just can't remember, for one particular condition in the pregnant ladies. So it was very popular in those days. But when they further saw the effect of the thalidomide, the children born to those mothers who were consuming thalidomide, they had absence of ear, malformed or absent thumbs, absence of arms, severe shortness of arms, dislocation of hip, malformation of ear, absence of legs, severe shortness of legs, or malformed thumbs. So basically, these were the effects that were seen on the mothers who were consuming thalidomide. So after that, the drug was banned because it was leading to a very abnormal situation uh, in the fetus that is being born. Okay, so cells do not stay still during embryogenesis. The mesenchymal cell, they undergo processes like condensation, cell division, cell death. If they'll undergo condensation, they'll form epithelium cells. And if they'll go cell division, basically mitosis, and they will form the limb mesenchyme. Interdigital cell death, which happens basically when a human is born, when the fetus is developing, so it has some kind of a web which is developed between the digits. So these webs must be dissolved, or must be degraded, so that we can move our fingers very use in very freedom. Because otherwise these webs will not let us move our fingers properly, it won't let us go beyond a certain limit. So obviously these must die. So this is the cell death that happens in the interdigital mesenchymes. Cell division in the mesenchymal cell leads to a formation of the limb mesenchyme, and condensation leads to the cartilage formation in the in the body. Then the migration helps in the heart mesenchyme formation, matrix secretion and degradation. It leads to the again cartilage mesenchyme formation. And growth forms helps in the formation of the fat cells. So if you can see, it is only a single type of mesenchymal cells, but depending upon the process it is undergoing, it can lead to the formation of different kind of the cells in that organism. The epithelial cell, again dispersal, it can delamination, and it can undergo shape change or growth. So again it will form different kind of things. It can involve in the neural part, it can get involved in the gut, it can get involved in whatever part it is required, and according to the process it is undergoing. Then again the thing come is the fate map. So fate map is basically nothing but it is just uh, we can say a map of different cells or different regions of a zygote or a developing zygote, blastula, gastrula, in which we tell that which cell led to the formation of what type of cell, which cell lead to that lineage. So basically it is fate map which tells us, which gives us whole of you can say an account of the cells that were involved in the development and what they formed in the final adult organism, or we can say the final organism. So fate map just tells us the fate of each cell. Okay, so that is why it is called as the fate map. Okay. uh, Next is the, it was directly observed in the concline and tunicate embryo. If I'm not wrong, these two organisms were used because the eggs were transparent. I guess I'm not very sure, guys. I have to read about that. I'll tell you in the next lecture. But why this concline and tunicate embryo was used, I just forgot the reason. But yes, these were the organisms which were used for the direct observation to understand the fate map, that if a particular cell is dividing and going somewhere, what it will form and what lineage it will lead to. uh, then different methods were used to study the fate mapping. One was a fluorescent dye, in which a dye is actually inserted in that particular type of the cells to to see the pathway it is going and what it is forming. Labeling is another method to study that, and again it was a genetic way to understand the fate of those particular cells, that where they are going and what they are forming. Okay, so these were the things that you guys have studied in the last lecture, and I hope you guys understood all of the things well. This was just the introductory week of the course, that what you guys will study in the coming lecture series. So, till now, everyone is clear with what we have discussed or what you guys had listened in the lecture series. Are you guys clear with what you guys had studied in the last lecture, in the last week's lecture? Because I have just given you an overview, we didn't went into that much detail, because this was just a recap of what you guys have studied in the last week. We are again not going into the details, but if you guys have problem with any part, we can discuss that. Anyone, any questions? In there? No. Germ cells is a different thing. Germ layers is a different thing. Germ cells are basically, uh, germ cells is a term which is generally used for the those cells, the ova and the sperms. These are formed from gonads, and gonads are formed from mesoderm. I'll just show it to you in the presentation. If you guys remember, when we, this was a, sorry, endoderm. So germ cells are basically the ova and the sperm, and these are formed from the endoderm. Endoderm is the third germ layer in the gastrula. So germ cells and germ layers are two different things. Germ layers are the ones from which different organs will be formed, and germ cells are the those cells which are actually the initial part, from where after fusion of which the zygote will form, and this zygote will undergo development to form the new organism. So I think you understood, Gayatri. Yeah, thank you. Okay, so should we move ahead towards the questions, the assignment questions? I hope you guys have solved all the assignment questions. Did you face any kind of issue on that? We'll discuss them anyways, we'll discuss them one by one. And if you guys faced any kind of an issue on that, we can discuss that also. Okay. So yes, I'm moving towards the question, and one request from to you all. Please do not speak up the answers. Write your answers in the chat box so that everyone can participate. I want to see that if everyone is on the same page and everyone is getting along what we are discussing. Okay, so I request you all to type your answers in the chat box. There's no hurry, you can take your time, but please respond so that I can understand that you are getting my point or not. You are getting something out of the course or not. Okay, so yes, we'll start. So yes, question number one. Morphogenesis refers to what? We have four options: multiplication of cells, embryonic growth, differentiation of cell, migration and adherence of cell in a certain way. I'll just see. Not getting the sounds. I have to check. Uh, Everyone answer in the chat box, please. Question number one, morphogenesis refers to multiplication of the cells, embryonic growth, differentiation of the cells, or migration and adherence of cells in a certain way. Differentiation of the cells. Just a moment, guys. I think there is some problem with the answer. Either I have taken it wrong or just a moment, guys. Uh, so according to assignment, the answer is migration and adherence of cell in certain way. Okay, so answer to morphogenesis is, but according to me, it should be answer C again. That's why I had to check. Uh, okay, differentiation cannot be. Yes, again, the answer is right. Migration and adherence of cells in a certain way. Differentiation cannot be morphogenesis. Differentiation is basically the deciding of fate, but morphogenesis is basically migration and what kind of a cell it is forming. So, migration and adherence of a cell in a certain way. Next question is observation of the expression pattern of a protein in a tissue section using a fluorescence microscope is what it is. Question number two, everyone. I hope the questions are visible to you guys on the screen. Observation of the expression pattern of a protein in a tissue section using a fluorescence microscope is what kind of an approach it is? It's an anatomical approach. Yes. Everyone. Okay. Are you getting the questions or not? Are you getting the answers? Because I've got answers from only two people, Shalini and Oli. Rest of them, are you guys not interested or you didn't get the question? Yes, come on, please answer. Didn't get it. Okay. So basically, the question, Nilanjana, here is if you remember, we have studied some approaches to study the embryology. So one of the approach was the anatomical approach. So they are asking if observation of an expression pattern of a protein in a tissue section using a fluorescence microscope. Basically, they want to observe a particular expression of a particular protein in a cell using the fluorescence microscope. So what kind of an approach it is? They want to ask you what kind of an approach it is. So we have been studying all of these approaches in the anatomical approach. So answer is anatomical approach. Did you understand it now, Nilanjana? Perfect. So rest all. What are your takes on that? Didn't you understand the question, or don't you want to answer the questions? Okay, no worries. We'll move towards the next question. Okay, so next question is, which of the following is or are central questions of developmental biology? Question number three, the central questions of developmental biology they are asking from the options. Just a moment, someone wants to join. Yeah. So they're asking which among the options are the is or are the central uh, questions of developmental biology. Now the catch here is the answer can be more than one. So you guys can guess anyone, more than one options for the question. I guess, but have a doubt in three as well. Shalini, you have a doubt in option third as well? Okay, we'll see the third option. So again, option one is correct. Why both of my hands are of the same length is a central question in the developmental biology. Third option says, why is the chicken neck longer than the mouse neck? So again, this is because what? What could be the reason for this central question? The reason is the environmental use. As I told you, the environment is what is affecting the organism to be that ways. It is the environmental cues that actually leads to the longer neck in the in one organism and shorter in other, because the requirement in the niche in which the organism is living, the requirement for that niche is the longer neck. But in case of another organism, it is not such requirement, so it won't develop that kind of a neck. These are some things which develop throughout the evolution. It is not a one day event, a one night event. It is developed throughout the evolution depending upon the requirements of that organism in the niche it is living. Did you get that, Shalini? Thank you. Okay. So the question answer to question number third is both options one and three. Okay. So next question is, why is the life cycle an important concept in developmental biology? Why it is an important concept in developmental life cycle is an important concept in developmental biology? What do you guys think? Shalini, is it the okay answer? You guys are saying it is four. Let's see. Uh, According to me, the answer is A, because it is a unifying theme applicable to all organisms. Obviously, life cycle means that only that it covers all aspects of the development from fertilization to reproductive maturity. That is right. That life cycle is that only. But important ये इसीलिए हो जाता है because it is a unifying theme applicable to all organisms. If we are trying to understand the life cycle, then we are trying to understand the development of all of the organisms. Okay, so answer for question number four is A, not B. Got it? Perfect. Let's move towards the next set of questions. So intestine is made up of cells from where? Intestine is made up of which cells? Question number five. Yes, right, right. Nilanjana, it should be endoderm and mesoderm both, because both the germ layers are involved in forming the intestine. Okay? So it is endoderm and mesoderm both. So answer to question number 5 is B, endoderm and mesoderm. Okay. Now the next question is, from the following, identify the statement that is true. So question number 6, you have 4 options. Now tell me which statement is true. Question number 6. And again, the answer to this question is multiple statements. Other people? Question number 6, everyone. Any other? Somi, Shalini, it is more than one statement. So go through the question once more and tell me which statements are right. No worries. We'll discuss this. So yes, in question number 6, the first statement says presence of gill arches in all vertebrate embryos is inconsistent with von Baer's principle, which is true. Completely false. It is not like that, because that is what von Baer said, that the general features in the larger group will come earlier. Okay, so next statement says similarity of embryos of diverse group at early stages supports Darwin idea of common ancestry. That is what we read in the first slide when we were talking about the evolutionary study. ठीक है, एवोल्यूशनरी पार्ट जब हम एनाटॉमिकल उसका स्टडी करते हैं एंड डेवलपमेंटल बायोलॉजी का, तो उसमें वो पहली स्लाइड जो हम लोगों ने देखी थी जिसमें वो बर्नाकल और श्रिम्प को कंपेयर कर रहे थे, तो उसने क्या बोला था कि सिमिलैरिटी ऑफ एम्ब्रायज ऑफ डाइवर्स ग्रुप एट अर्ली स्टेजेस सपोर्ट्स डार्विन आइडिया ऑफ कॉमन इंसेस्ट्री. So this is a right statement. Next is bat wings and human hands are homologous structures. It is right. Bat wings and butterfly wings are analogous structures. ठीक है? So obviously again these are statement number 2nd, 3rd and 4th. All the 3 statements are right. Okay? Did you get that? I hope everyone understood. Next question. How do human birth defects help developmental biologists? Question number 7, everyone. Question number 7. How do human birth defects help developmental biologists? Okay, so answer to question number 7 is identification of underlying mutations help us identify genes involved in human development. Okay, so Nilanjana, why option 2 cannot be correct is birth defects help us identify the expression pattern of human genes. This is not the case. This is not telling us the expression patterns. This is not something which is getting affected at the protein level. It is getting affected at the developmental level only. Okay. So it is not telling us the expression patterns, but it is telling us which mutation is affecting which part of the development of human. Okay, Nilanjana? I hope you got the answer. So second cannot be the right statement. Okay. Next question. Question number 8. What does this says? The term teratogen refers to an environmental agent or chemical that what? Teratogen term किस लिए यूज की गई है? क्वेश्चन नंबर 8 का आंसर great. Good guys, good. You're performing good. So yes, the answer is to the term teratogen किस लिए यूज की गई है? So the term teratogen refers to any environmental agent or chemical that affects embryo development. ठीक है? It affects the embryo development. Okay, so question number 9. Why did Conklin choose tunicate embryo for fate mapping? Why did Conklin choose tunicate embryo for fate mapping? I told you in the series also, I forgot the reason. So now you guys tell me the reason. Again, the answers are more than one. So tell me. Answer to question number 9. Mm-mm. Who was the guest? Who wants to join? I can't see anyone. Yes, the answer is 2 and 4 because the cells are pigmented, which helps us to understand the fate map better, and the cleavage is invariant, so we can understand it better. Okay, next question. Which one of the following is most suitable for lineage tracing of cells that form very late during development? For example, the bones of frog jaw. बताइए, आज हमने डिस्कस भी किया लास्ट इमेज जो मैंने आपको दिखाई थी. Question number 10, everyone. Answer to question number 10. Question number 10. बताइए सब लोग. Battery is getting low. हाँ जी. Okay, so the answer to this question is D. Just a moment, guys. Which is this guest that I cannot see? Oh, sorry. The screen sharing is stopped. Sorry for that. Is my screen visible to you guys? I hope it is. Thank you. So yes, the answer to this question is transplanting from a transgenic embryo carrying a reporter gene. Okay, this is what the answer to question number 10 is. So with this, we finish the assignment questions. I hope you guys understood the questions. You guys understood why the answers, what they are. And now let's move towards the questions that I have brought for you guys. The very easy questions just to brush up your lecture. Okay, so let's move towards that. So the first question is, the product of fertilization is called what? I've been using that term more often in the lecture. Okay, perfect. Yes, the answer is zygote. The answer is zygote. Yes, right. Next question, which process forms the three germ layers? Perfect, perfect. So it is the gastrulation which forms the three germ layers. But next question. The nervous system develops from what? From which germ layer? Badhiya, badhiya, badhiya. It is the ectoderm which give rise to the nervous system. Good. Next question. The notochord is derived from. Notochord is derived from. Yes, it is derived from mesoderm. Good. Badhiya. Next question is, endoderm mainly forms. Wow, very good. It is the gut lining that is mainly formed by the endoderm. Next question is, cleavage consists of. What happens in cleavage? Yes, it is the rapid mitotic divisions. It is because here this cell is not growing, it is just dividing and dividing and dividing whatever it has already. But in case of the mitosis or meiosis, what happens is, cell undergoes the full cell division cycle. First it will grow, then it will divide, it will grow and it will divide. So size of cell remains same, but in case of cleavage, the size of the cell keeps on decreasing because it is not growing, it is just keep on dividing, dividing and dividing. It is not undergoing the full cell division, it is only undergoing the division part, it is not growing. Okay, so that is the main difference between a cleavage and a division. Okay. Next question is, a blastula is. What is a structure of blastula like? Yes, everyone. Question number 7. A blastula is. Okay, so only Nilanjana got it correct. It is the hollow ball of cells. Did you forget a blastula has a blastocele? So it is a hollow ball of cells. It is not a solid ball. So only Nilanjana got it correct, guys. Please थोड़ा सा बारीकियों पे ध्यान दें और प्लीज थोड़ा सा स्ट्रक्चर्स को याद करने की कोशिश करो so that you can get it कि which structure is like what. ठीक है? Okay, next question. A holoblastic cleavage occurs in what type of organisms? That's okay. Biology is all about terms. If you remember the terms and if you read the question carefully, you will never get wrong in biology. So question number 8. Holoblastic cleavage occurs in. I've got one correct answer. Other people also answer. Yes, the answer to question number 8. Yes, it is amphibians. And meroblastic cleavage occurs in. Other people don't want to answer question number 8 or 9. First, all of you please answer for question number 8. Holoblastic cleavage occurs in. Yeah, it is amphibians. Yes, right. And amphibians include what? What kind of an organisms comes under amphibians? Can you give me an example of amphibians? Frog, right, right, right. Next question. Meroblastic cleavage occurs in. Yes. Other people please answer. Meroblastic cleavage occurs in. Yes, yes, it is birds. It is the birds in which the meroblastic cleavage occurs. So with this, I think we are finished for. No, no, no. We have some more questions. Sorry. No, no, no. We are done for the day. We have these are the only questions that I got for you guys. Next time I'll bring more. But for today we are done with the questions that I brought. Now if you guys have any confusion, if you guys want to ask anything, you can ask me. I'm giving you guys time. You can ask me if you guys didn't understood anything from the lecture, if you guys had any problem with any of the questions in the assignment or from the questions that I brought, then you guys can ask. Any problem you face today, you can ask that also. So all of you understand Hindi in the group? Because I was using somewhere Hindi language also. So I hope you all of you understand Hindi. Gayatri, you don't understand Hindi. That's okay. So anywhere if you don't understand when I was speaking in Hindi, you can tell me. I can repeat that part. Did you face any problem in the lecture somewhere? Any questions, anyone? Okay, perfect. You can ask me today, otherwise, you can ask in the next class also from this week's part also. There's no issue on that. You can ask me questions then also. And if you have today some questions, if you didn't understand any part, then we can discuss it now also. That is totally up to you. No issues with that. No questions, anyone? Yes, sir has mentioned already on his page, and if you will see in the lecture one notes, in the initial slides only, he has mentioned the book from the writer Gilbert. So that is the best book you can refer for developmental biology. This is the best book that can teach you development biology the best. No other book uh, works better than Gilbert. So you can refer to that book, Nilanjana. Okay, so holoplastic cleavage is that cleavage in which whole of the cell, the zygote cell, undergoes cleavage. But in meroplastic cleavage, only some part of the cell undergoes cleavage. The rest of the part doesn't undergoes cleavage. So this is the difference between a holoplastic and a meroplastic cleavage. I hope you got that, Gayatri. So basically, the main objective of the session is, if you have any kind of a doubt, if you didn't understand anything from the lecture series, so you can ask it here in the session. Otherwise, we'll discuss what just a quick recap, or we'll discuss all the questions that you brought. Otherwise, we'll discuss the questions of assignment and some of the questions that I will be bringing. So that way it can help you in getting the lecture better. The whole of the objective for the session is that only. Any other thing anyone want to ask? Done for the day? Okay, guys, if you don't have any other questions to ask, then you guys can leave the meeting. We'll stop the session now and we'll meet you next week, same time, 6 p.m. Tuesday. You're welcome, guys.